We're going to look at the Unit 3, Microscopy, Biology Near 4. This unit, in microscopy, we are going to describe the main features and functions of the components of a light microscope. State that magnification is the increase in the object. We know that with microscope we are able to visualize the unseen objects, those which are not able to be seen by our naked eye. So those objects have what we call magnification. We distinguish between magnification and the resolution. Resolution is the ability of the microscope to show two objects as separate. We'll see what we call actual size of the specimen and the image. We also have calculations whereby we'll be calculating the approximate size of different biological structures using an appropriate unit of measurement. We'll see the advantages and disadvantages of using an electron microscope. Lastly, we'll see the advantages and disadvantages of using scanning electron microscope. Let's start with compound light microscope. All of us, we are familiar with microscopes from our schools, from the hospitals, from the health centers, and even the other institutions of scientific research. Those microscopes we know are those called compound light microscope. This is the optical microscope, often referred to as light microscope. This is the type of microscope which uses visible light and a system of lenses to magnify images of small samples. It uses transmitted light for observation. However, microscope uses specific light characteristics for specific samples, such as transparent specimens and samples that do not pass light. Light source is focused on the specimen, the condenser lens. It then enters the objective lens, where it is magnified to produce a real image. The real image is magnified by the ocular lens to produce a virtual image that is seen by the eye. Unfortunately, as we are online, we don't have a microscope, a physical one, so that we can manipulate it as to look at those different parts. But I think that in your schools, you have ever been with the microscope and now you have seen all the parts. Even though we don't have it now as physical, I tried to search for a diagram, a drawing, or a picture of a microscope. First of all, we see the ocular lens or eyepiece, and through it we focus, we see the objects. We have diopter adjustment because you adjust when you look at their inside. You are going to adjust the image so that it can be closer or far away from you when maybe it is with too much of light. You adjust, not you adjust the object not being close, but it can be far. You see the head on which we have ocular lens. You have the objective lens. Objective lens, these are for magnification, and we have different magnifications. We start by 10x, 40x, 100x. Those are different magnifications. We're going to meet them during our exercises. You see there, arm carrying a handle. When you carry your microscope, this is the press where you touch with your arm. It's called carrying handle. Your hands should be when you are to lift. Down you see the stage creep on the aperture. Down you see the diaphragm and the illuminator as the source of when you connect to the power. There you are going to switch on, or that illuminator is in a glass and it is there from which light comes, and it is that light which is going to facilitate us to look at the object from our ocular lens. There you have mechanical stage. Yes, at the mechanical stage, you place your specimen on a glass. Suppose you are going to start the cells in our blood, the sample drop of blood on the cover or on the glass, and you put the cover, then you place on mechanical stage. Then you can adjust. So force adjustment, it means it's going to be up or down according to your weight. You can now adjust towards that illuminator so that light may on your specimen. You can adjust towards up so that light may not tense on your sample. There you find the stage controls. Down you see the base. The base where also your hands. At the same time, the arm when you are lifting or you are carrying a microscope. Then you are seeing the brightness adjustment there down. You can now turn. So when you turn, you are going to clearly see the specimen which is placed on the mechanical stage. You find the light switch. After connecting the power of the electricity to switch on, or you switch off when you finish your working. These are the parts of a microscope. Let's now go with magnification and resolution of a compound light microscope. Magnification refers to increasing the apparent size of the object, while resolution of a microscope is the ability to show two close objects separate. Magnification of an ordinary light microscope is about 100 and 1,500 times. Magnification by formula is called measured size over the actual size. Also is equal to the size of the image over the actual size of an object. Here we have an example of calculation. Calculate the magnification if the size is 5 micrometers and the measured image of the specimen has the size of 4 micrometers. By the answer, you make the size of the image and the actual size in the same units by converting to micrometers. This is done by multiplying by 40 millimeters by 1000, so that 40 millimeters becomes 8,000 micrometers. These are the standard units which are used in those smallest or those invisible objects. So magnification is equal to the size of image, which is equal to 40,000 micrometers, over the actual size of the object, which is 45 micrometers. That is equal to times 8,000. This is the magnification. You know, magnification has no units except that sign of multiplication which starts, and sometimes it is like to place at the end. Instead of writing times 8,000, you can also write 8,000 times. Note that the magnification specimen under a right microscope is created by multiplying the magnification of the objective used to that of the eyepiece. For example, 10 times this is an objective, 10 times eyepiece is equal to times 100. Exercise are there, but you are going to understand this. Let's look at the electron microscopes. Electron microscopes are now host the compound light microscopes because, like here in what you're familiar with are compound light microscopes. I mentioned those in our school. For light microscopes, they are rarely found here in Rwanda. I never found one. Is the progress development of the country. It may be there, but I've never been lucky to see it. Microscopes used to study details of internal structures called the ultrastructures of the cells. The two types are transmission electron microscope and electron microscope. More transmission electron microscopes can distinguish objects as far as 0.2 nanometers. Remember that nanometers is equal to 10 power 9 mega 9. This can produce clear images magnified to 250,000 times. That it is powerful than that. Can use clear images magnified 250,000. Electron microscope of accelerated electrons as a source of illumination. Electrons beams have much smaller wavelength than light rays and therefore have a greater resolving powers, can produce high effective magnifications in light microscopes. If you understand that there is a big difference, you never compare the two. Electron microscopes are far away more to compound light microscopes. Here we have the image of the electron microscope. I never image in my life. These are very advanced. I say that perhaps today we have them here in our country, but if they are there, the institutions I have never entered in till now. The transmission electroscope a beam of electrons is emitted and processed to form electron micrograph on a fluorescent screen. You see now the parts of an electron microscope. But surely on you are able as students of high school, You are not going far with electron microscopes, except microscope are easily found on your reach. I'll continue with electron microscope. Is scanning electron microscope used to produce of surfaces of the specimens. Electrons are reflected from the surface of the specimen stained with a heavy metal, and this enables the scanning electron microscope to produce images of all specimens, cells, tissues, or even organisms. Here we have also the image of scanning on microscope, which is here. I hope that with advancements in science, we are going to act. Advantages of the Overlite microscope. Remember that light microscope is the one we are familiar with in the places with hospitals, schools, etc. Electron microscope has higher resolution and is therefore able to have a higher effective resolution estimated at up to 150,000 million times compared to light microscope, which can show a useful magnification only up to 1,000 or 2,000 times. You see, there is a huge difference. This is because a light microscope uses a beam of light with a longer wavelength, while electron microscopes use beam of electrons that have a short wavelength. But we have disadvantages. These microscopes, even though they are most powerful, not good sometimes. Because first of all, expensive, extremely expensive, and the maintenance costs are high. Preparation is often much more technical, requiring special training, high level of samples must be dead to high radiation and are placed in vacuum so that people observe living specimens. It is not possible to observe colors because electrons do not possess. The image is all black white, even if sometimes artificially to give a depth. They require more training and experience finding artifacts that may have been produced during the sample preparation process. Using for some advantages of electron microscopes, extremely expensive and also requiring high density. Let's now compare between light and electron microscopes. Both light and electron microscopes form larger or magnified and more detailed or higher resolved images of small objects or small areas of larger objects. Electron microscopes are used in biology study, research, and medical sciences, particularly histology, material sciences such as metal and other aspects of science. Be carefully prepared using techniques for both the equipment and the sample, including staining and mounting. These are some comparison points between light and electron microscopes. They magnify, they give resolved images used in the same institutions. They may used in medical studies, science studies, medical sciences, etc. Now let's enter the because we have seen the concept of microscopes, compound light microscope we are familiar with, and the light we are not familiar with. Now we have seen the concepts of magnifications, resolutions, image size. Now let's be familiar with short calculations which are involved in microscopic studies. Examples, calculations about microscopes. Suppose that length per cell is 150 micrometer. Size of the cell image with the microscope, magnification of multiplication 50. Give your answer in micrometers, and I want you at each example to give you some one so as to understand the question and look at the answer, how the way they are calculations which are held there, then make sure you understand the process. And when you allow me to, I will continue. First of all, look at working and make sure you understand, because some more exercises are you understand the working. You see the low length of the cell, 150 micrometers. What is the size of the cell image with a microscope magnification of 50 times? Here we are given magnification and we are given the object size, the U length of the cell. And the image size is equal to magnification times object size. Remember that 1 micrometer is equal to 1,000 micrometers. That's why the image size is equal to the magnification of 50 times the object size of 150 micrometers, which is equal to 7,500 micrometers. And you should now divide them by 1 to get the answer into micrometers. Any question here?
No.
Okay, thank you. Example number. The red blood cell is 7 millimeter under a microscope magnification of 1,000. What is the real size of the red blood cell? Give your answer in micrometers and nanometers. 1 millimeter is 1,000 micrometer. Millimeter is equal to 7,000 micrometers. Again, the formula, the object size is equal to the image size over magnification. That's why you find the image size, which is equal to 7,000 micrometers, over the magnification of 1,000 until 7 micrometers. So you are going to convert 7 micrometers in nanometers. You know that nanometers is called 10 power negative 9. And micrometer is 10 power negative 6. That's why you have negative 6 and 9, 7 times 10 power 3 nanometers. Is it clear? And the conversions?
Show it.
Okay, thank you. Look where at the working. Make sure you understand all the process of work before continue. Before we continue, make sure you understand the whole working process. Another example. What magnification is needed to produce a 10 millimeter image? 10 millimeters image of a cell from a specimen cell of diameter 125 micrometers. Now we are required to calculate the magnification. Magnification is required. You know that definition says that magnification is equal to the image size over the actual size. Remember that 1 millimeter is equal to 1,000 micrometers. So they convert 10 millimeters into micrometers by timing it with 10,000. No, sorry, with 1,000 to get 10,000 micrometers. Look at the working also. And I want to know you understand it so that we continue.
Teacher, you can continue.
Okay. Now get ready with exercises. Your notebook, your pen with your calculator machine ready now. Just calculations as simple as those simple calculations you see here. Now we are going to start with exercises. Are you ready?
Yes.
Yes.
Okay. A paper and a pen around. We start. Here we have the size of the image. We have magnification. We are required to calculate the actual size. Assume that the student is drawing at the same scale with the magnified image.
The way I have done this question, they told us that we have eyepiece magnification of 10 and objective lens magnification of 40. Then what we needed to first do is to calculate total magnification. We have learned that to get total magnification, you take eyepiece magnification times objective lens magnification.
Thank you.
Then I take total magnification was equal to 10 times 40.
Go to
And
400
which was equal to 400
times.
then times. Then we know that magnification is equal to image size over actual size of the object. Then we will find that object size will be equal to image size over magnification.
Yes.
Afterwards, I find that object size is equal to 120 millimeters over 400 times,
Very good.
which finally give 0.3 millimeters, which is equal to 300 micrometers. Thank you.
A big crap. Number 2 is coming. We have a cell diameter 0.00090 meter. What is its diameter in the standard form? What is its diameter in micrometers? Number 2, please. 1 minute.
Thank you
Okay.
for the question, which asked to put that diameter of the cell into standard form.
Yes.
I found that it could be 9 times 10 power negative 5 meters.
Very
Thank
good.
you.
A big clap. Bright student as you are. What about the others, please? The rest. Now let's go to B. This diameter into micrometers form. First of all, you have the standard form, 9 times 10 power negative 9. It is that one we're going to convert into the micrometers. You know that one micrometer is equal to 1,000 millimeters. Means you get the answer into the standard form times 1,000. And you know that 1,000 is equal to 10 power 3. Yes or no? If Farine says that the standard form is 9 times 10 power negative 9, for B to micrometers, it will be to 9 times 10 power negative 9 times 10 power 3. And the answer will be 9 times 10 negative 9 times 10 power negative 6. Is it okay? Have you tried to find the same answer as me? These are calculations from senior 1 in physics. I remember 2 units, standard units, converting. Any question? Others, are you sleeping? Tell me if you hear me, you understand what we are doing. If you have any question, you raise your hand, you ask. Maximize your opportunity here now. May I continue, please? Unmute yourself when you want to talk. Number 3, a specimen is 6 micrometers wide. Calculate the width of the image in millimeters under a magnification of times 200. Think about the working. When you were asked the width of the image, it is the same as to ask you the image size. You have the real size of the specimen, which is equal to 60 micrometers. You also have magnification. What you are asked to calculate is the image size. And you know that the image size by the formula is equal to magnification times the real size. Now I am helping you. Okay, do the rest. Apply the formulas now. What is the answer?
The answer is 12 millimeters.
Very, very good. 12 millimeters.
Yes.
Now, the last exercise towards the end of this unity today, a series 3 times 10 power negative 5 meters wide. Calculate the width of the image under a microscope magnification of times 200. We quote your answer in meters, in micrometers, or written in the standard form. Try it in 1 minute. The same working as the previous one. Apply the formula immediately. This is the same as number, the previous number we have just finished. Nikuze, I see you are done.
Yes.
Tell us about your working.
Thank you. What I have done, we know that to calculate image size is you take magnification times actual size, and
Yes.
we had magnification of 200 times.
Yes.
Then and actual size of this of the cell, which was equal to 3 times 10 power negative 5 meters. Then
Yes.
what I have done is that I took 200 times 3 times 10 power negative 5 meters, where I got 6 times 10 power negative 3 meters. And we know that, and then I changed this 6 times 10 power negative 3 meters into micrometers. Finally, I get that in micrometers, its image size is equal to 6 times 10 power 3 micrometers. Thank you.
Okay, big clap. Cool. Any question about this topic? the concepts we went through and these calculations. Any question? If I have a question, please be bold. You express it. Oh, I see. There is no question. When you don't talk, there is a consent. This is a saying. It means you understand everything. I'd like to thank you for your kind attendance and participation today in this Unit 3 of Microscopy Senior 4. Next time, that is Wednesday afternoon at 3 p.m. We'll start with unit number 4 about cells and tissues.
Oh, genius!